So this is our original artwork. Apologies for the wrong date, but I thought it was more informative than the one with the reschedule across the front. So first and foremost, we need to extend a big thank you to Claire for giving us this presentation this evening. It's always demanding, and I'm sure that life has become trickier for Claire in the same way it's become trickier for everybody under our various lockdown constrictions. So let's go on with the fact. Note down the bottom here, this webinar is recorded. It's already recording because that was an easier way for me to do it and stops me from forgetting. It will be edited up and ultimately put on the Dogs Victoria website for the information of other people down the track. We've already spoken about the rules, but let's go with them yet again. Please make sure that your microphone stays muted to minimize interruptions. Use the chat function so that we can respond to your questions, and I might let Claire explain how you might want to use the Slido option because she has far more expertise in that area than me if you need to ask questions. So a little bit about Claire. Here she is with a toddler baby. Chair is in fact the professor of the computational biology and animal genomics at Sydney University under the Animal Sciences Cluster Chair in the Faculty of Science, School of Life and Environmental Sciences at the University of Sydney. Claire is also a member of the World Small Animal Hereditary Diseases Committee, an expert panel member on dogwellnet.com, and director of the Royal New South Wales Canine Health and Welfare Charity. Is that still the case, Claire?
It is.
It is, right. I wasn't quite sure whether you were still continuing on with all your responsibility. And most of all, Claire is also a passionate dog lover and enjoys her beautiful tollers. So thank you, Claire. I think we'll now go back to my smiley face. I hope it's smiling. Admit a few more people, and I would like to hand over to you for this evening's seminar.
Thank you, and thank you so much to the Dogs Victoria Education Committee for inviting me to come and talk to all of you guys. It's very exciting to talk to people about dog genetics. I love, my most favourite thing in the world is talking to people about dog genetics. And unfortunately, not all of my students are as enthusiastic about it as you guys are, but still some of them are. And so, you know, that's exciting too. So it's lovely to get to talk to people about genetics, even better when it's about my favourite animal. And so I'm going to share my screen. Margaret suggested I talk to you about Slido, and to be honest, I have like 5 minutes more of expertise in it than Margaret does. But the Slido will allow you to put in a question in real time, and then hopefully someone will be monitoring those so that we can cover most of those questions at the end of the presentation. And you can, the other thing you can do in Slido is you can put a like on the question, and that will bring it up to the top. So that might be another thing that you want to do. If there's a question there that interests you, you can push the like button on the question, and that will make it float to the top of the screen so we see which ones people think are more important. So if you have that Slido screen open at the same time as this screen, then all will be good. So what I'll do now is share my screen, and I'll see how I go doing this. Hopefully it will let me do that. Oops. And hopefully it will let me go from the beginning. Here we go. If the window over here is in the way, you can always just move it around. There's a little button in the corner with the smallest little horizontal bar, and that will make that really small if you want it to go away entirely. And I'm going to do that so that I don't look at my face and I can talk better to my slides. So what I was hoping to do was just to begin by covering some basic genetic principles as they apply to practical dog breeding. And I'm not going to go through the whole meiosis thing. We're going to stick to the things that are more likely to be relevant to you. And so we'll have a brief introduction to general genetics principles to sort of get us on the same page. And then after that, we'll go to the questions that have been put in through Slido. And then after that, we can take other questions from the floor if those should arise. And hopefully Margaret or someone else will be monitoring the chat so that if there's some urgent thing that you want to draw our attention to, I can cover it in real time if through the chat, but someone might need to tell me about that. So I'll be just seeing my slides in the share screen. So someone will need to tell me if there's chat that needs to be attended to.
I can do that for you, Claire.
Okay, so I'm going to just minimize the little viewer thing for me. All right, so here's a beautiful picture of my girl Trouble and some of her puppies. And Trouble, as you know, has been a little bit unwell lately. She has gastrointestinal problems, which are not very pleasant. And so we're just going to cover, and I know, first of all, I know people always tell me, oh, that's not a very good example of that breed, but unfortunately, you know, you have to get what you can get from a stock photo, except for the dog on the end, which is my old dog Bern that I brought over from America. He was my first holer. So I guess to begin with, the first thing to understand is that not everything is inherited. And so there's no point selecting against things that are not inherited in a breeding program, unless it's for the welfare of the animal. So you don't want to breed from a sick animal because it's sick and it would put stress on the welfare of the animal. But in terms of genetic improvement, if an animal's sick, that doesn't necessarily mean it's an inherited problem. And so selecting against an animal, like from a genetic standpoint, you need to be sure that what you're dealing with is something that's inherited. So how do we know when something is inherited or when it's not? And so I guess the first things to understand are that when things are inherited, they more often appear in members of the same family. So when we say that a trait runs in the family, so if, for example, in my family, we have red hair that runs in our family, then one might presume that red hair is inherited because we've seen it in multiple members of the same family. So what makes a family important is that they share genes, and we'll talk more about this in a little while. The other animals that share genes are other members of the same breed because they're all sort of like an extended family. And so members of the same breed are more likely to share genes or gene variants than members of different breeds. And so if we see a trait that's regarded as a breed characteristic or a breed problem, then that's more likely to be inherited than something that's across many breeds and doesn't seem to be particularly focused in families of any kind. There are, however, some exceptions to that I'll talk about later as well. Oops, sorry, I've disconnected my keyboard, so I have to use the one on the laptop. Okay, so even with congenital defects, not all congenital defects are inherited. We need to ask, is there one puppy in the litter or more than one puppy? Is it one litter or more than one litter? And is it one breeder or more than one breeder? And actually, someone contacted me recently regarding a situation where there were various neurological defects being thrown in multiple litters with the same breeder, and they were presuming it was inherited, but there are also infectious diseases that can be passed maternally. And so it's possible that that was an infectious disease. So you need to rule out that you're dealing with an infection rather than a hereditary defect. Now, in the case of this puppy here, which is a Coma Defect puppy, the real name for that spondylocostal dysostosis in miniature schnauzers, in this case, these puppies appeared in multiple litters from the same breeder where the mothers of the litters were related to each other, but it wasn't the same mother each time. And it so happened that the fathers of the litter were also related to each other. And so this did in fact end up being an inherited defect, and it's inherited in a recessive manner, which we'll talk about more in a little bit. So it's more likely to be inherited if you see it in more than one litter, and ideally more than one dam from the same breeder, or even more than one breeder in the same breed. So we shouldn't waste our precious selection pressure on things that we don't know to be inherited. So oftentimes many of us will see that sometimes you'll get a defect in a puppy. Sometimes those can be caused by things that happen during pregnancy. Sometimes they are inherited, but they might be extremely rare. And so until you know that it's inherited, it's best to not use that as the basis of excluding a dog from your breeding program. So when we make improvement in our breeding program, all of us have in our mind some vision of what we see as the perfect outcome of our breeding program. And one thing that I've noticed as a geneticist is that nearly everybody has a slightly different vision, even when, you know, if you ask non-dog breeders about what dog breeders are breeding for, and everyone says, oh, they're breeding for the breed standard. Well, they may be, but they may be breeding for different aspects of the breed standard and putting pressure on different aspects of the breed standard. So some person might have a thing for head shape, and another person might be really into top lines, and another person might be all about temperament, and another person might be all about health. And those different slight differences in people's breeding goals mean that they all select along a slightly different trajectory on their progress towards their vision of the perfect breeding program. And I think that's a good thing, because even though we all want the breed standard, with each of us having slightly different goals, that allows dogs to maintain a lot more genetic variation than what people outside the dog world give them credit for. So we do get to maintain a lot of genetic variation, because not everybody does have the same. Even though people say we're all breeding for the standard, we all want this, we all want that, actually that's not true. And more often than not, people will breed the dogs they have in their yard, and they'll make excuses to breed them, rather than they'll try and correct their problems rather than ditching their dog and starting again with another one. So the other thing to understand is that many, many traits are not simply inherited. So many of us will have heard about recessive genes and recessive inheritance. And these are really the low-hanging fruit when we're selecting for things. They're the easy things to make tests for, and they're the easy things to control in the breeding program. But by far, the vast majority of things that we're interested in are not simply inherited. And so I guess if we think about hip scores, temperament, litter size, breed height, all of those things, working ability, all of those things have very complex inheritance that you can't just make a genetic test and suddenly everything's perfect. So you need to be aware of that. Okay, so generally the rate at which we make genetic improvement depends on a few different things. The first really important thing is to focus our attention on the things that are heritable, as we were saying earlier. If it's not inherited, then you'll be putting all this selection energy into something that doesn't yield any benefit in the next generation. The other really, really, really important concept is the concept of selection pressure. It's a limited resource that you have to use. So when you breed a litter and you want to choose an animal or animals from that litter to take forward in your breeding program, you only have a limited number of animals that you can choose into your breeding program. And so how you choose that animal depends on the amount of emphasis that you put on different things that are in your vision, your breeding goal, your breeding objective. And so the more things that you have in your breeding objective, generally the less progress you'll make in any one thing. And I guess historically, people tend to focus on things where they see an improvement. They're less likely to persist if they don't see an improvement in the next generation or two. And so an example of this might be, you know, I have friends in retrieving who have tried to bring the working ethic of working Springer Spaniels into their showline Springer Spaniels, and then they've not persisted with it because in the next generation, the puppies are not particularly good at anything. And so, you know, long term, that might be possible, but you're not likely to see anything you want in the short term. So one thing is, how many animals are you going to choose from those that are available? Because you can make, if you imagine a group of people in a room, and let's make it easy and say they're all women, then each of those people will be a different height. And if you made them stand behind a little marker that described their height, you'd see a few people that were really, really short, I should say over here for you, and a few people that are really, really tall, but the majority of people will be kind of in the middle. And we call that a normal distribution. But the effect of that is that if you only select one animal from all of those animals of different heights, then you're going to make a much bigger improvement than if you select half of them, which will only bring you back towards the middle. So in order to make a genetic change, you always make more genetic change if you select fewer animals from a lot that are available. The other important thing is how much emphasis you put on each trait. So if you do your first selection, you say, oh my goodness, I can't condone a toller that has a black nose. Then immediately, if there are black noses in your litter, then that, you know, that's where you've basically put all of your selection effort there. So whatever, usually the first cut is where most of your pressure is going to go. So you've got to really figure out for you what's important. The other thing that's really important is stability in your breeding goals. So if you say, okay, this time I'm going to select for flesh-colored noses in my tollers, but okay, I've got those now, so I'm not going to care about what color nose the next sire has, but next time I'm going to have the ear shape be just perfect, then that makes it hard to make progress overall. You tend to go 2 steps forward and 1 step back when you do it that way. So I often hear people online, I know there's been a bit of sales pitching around the veterinary surgeries around Australia selling pen hip, and a lot of people would have you believe that traditional hip scoring like has been done through the ANKC is not effective. But actually it has been effective, and there is a gradual improvement in hip scores across all breeds that have applied hip scoring using X-ray evaluation of the hips by whatever method that people have. So that's just to show you that even when a trait is inherited in a complex way, you are going to make improvement if you persist with selecting on that. The improvement may not be as quick as everyone would like to see, unless you put all of your selection pressure on hips and nothing else. If you didn't care about temperament, if you didn't care about conformation, if you didn't care about color or working ability, then you could have, you know, excellent hips in every dog in no time. But the reality is that in nearly everybody's breeding program, we're concerned about more than just hips. And so I accept that we have this gradual improvement in hip scores, and I actually don't want to see it go hard personally, because to me that would just become another selection of extremes, and you'd end up going down the road of, you know, other problems that we have in dog breeds that have extreme conformation. Got to remember to push the right button. Okay, so in order to make genetic improvement, you want a clear vision of what it is you want to achieve. The more traits that you have, the lower the rate of improvement you'll get in each one in any period of time. And you only have very limited selection pressure to spend. You've got that one animal possibly that you're keeping, And so you need to make sure that you don't waste that selection pressure on things that ultimately are not that important in the big scheme of things. So always keep focused on things that are heritable and things that are really important to you and your breeding program. All right, so next I want to talk to you a little bit about chromosomes and genomes and inheritance. And a lot of you will kind of know this, but some of you may not. And so inheritance is made possible by animals passing chromosomes from one generation to the next through eggs and sperm. And one of the things that makes dogs an excellent model for mapping genetic things, and why a lot of scientists like working with dogs as models, is because they have lots of chromosomes. And when you have lots of chromosomes, it means that the genes are more separated from each other and easier to find in the greater DNA of the animal if you're looking for a specific gene. So if we were to look at these chromosomes, the picture down here on the left, I don't know if you can see my little, oh, it's going to go down here on the left. This is shown as an X, but actually dog chromosomes are not very X-like. They tend to just be like a sausage. But if you were to unravel that X, if you want to unravel one of these, it's wound up really, 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 really tight into a spiral. And as you unwind it, it's like a ladder. And as you unwind it further, eventually you get down to these A's, C's, G's, and T's, which make up the genetic code of the dog. And each dog has about 2.5 billion, that's 10 with 9 zeros, 1 with 9 zeros after it, letters of DNA. And before the age of supercomputing, we really were not able to do very much of any impact in mapping genes or understanding how genes work, because it just wasn't physically possible to look through or understand what was going on in those 2.5 billion letters of DNA. So dogs have nearly twice as many chromosomes as people, and some people can actually tell you which chromosome that is by looking at it, but I can't. Oopsie, sorry. Are you still there? My screen share's gone haywire. Alrighty, so what happened then? I clicked on something I shouldn't have done. Here's where we were. Alrighty. I keep forgetting that I'm on the laptop and I keep trying to use my desktop keyboard. Alrighty. Just kind of up the thing. Can someone give me a thumbs up that everybody's still there? No? Yeah, good, excellent, thank you. Alrighty, so just to get some terminology down that geneticists use that everyday people don't really use very often. Most people will have heard of a gene, and in a way, the concept of what is a gene has changed quite a lot in the last 20 years. And to be honest, I'm not 100% certain that I would completely know how to categorize a gene, given all we know about the way our DNA works now. But traditionally, a gene was a specialized segment of the DNA that made that had the instructions to make a protein that was then used in a body to do stuff. So I guess milk is a protein, egg white is a protein, cartilage is a protein, even bones have protein in them. But essentially, it's a place in the genome, a genome being the entirety of the DNA. And so another word that geneticists use to describe this is a locus. So a locus is just like a X and Y coordinate on a map. And one of the things about having a reference genome means that we're all talking about the same map. So that when I say, you know, the MC1R gene, which codes for the red or yellow hues in Labradors, for example, is on chromosome 5, then I'm talking about the same chromosome 5 that another geneticist is. So at that address on chromosome 5 that has the gene that we're interested in, so say it's MC1R, which is the E locus, then at that place, everyone says the dog has the gene for this or the gene for that, but actually most of us have all the genes for most things. It's just that the genes are not quite the same. So I used to always say to my students, it's like having a street address, and at a certain house number on that street, you might have a vacant lot, you might have a house, you might have a block of units, you might have a hotel. So there are many different things that can live at that address. And so the genes might be, well, there are 2 houses that, you know, that could live at that address, but one house is not quite the same as the house next door. And if it were that address and it was the other side of the street, if it was exactly the same house, that would be like the genes being the same, but actually we have a lot of individual variation in the way our genes are. And so, for example, in that MC1R gene, in dogs, it's very common that dogs that have a red or a yellow coat color have 2 copies of a gene that makes the protein not be made completely. And so that protein doesn't work now. Okay, so for most chromosomes, animals have 2 copies of that chromosome, except the sex chromosomes, which are a little bit different. And for that, that pair of chromosomes, we'll talk more about this in a moment, you get one from mum and one from dad. So in a population, let's talk about another coat color gene, the gene TYRP1, which codes for the brown coat color in a lot of breeds. There are at least 5 different variants that cause brown coat color. And so your dog could have not the black version, but it could have 2 different brown versions and still be brown. But the tests for each individual brown version may make it look like the dog's black. But there are different versions of that gene that all code for brown. And if you have 2 versions of the gene that code for brown, you'll get a brown dog. All right, so if the 2 variants that an animal has, one from mum and one from dad, are precisely the same, then the animal is said to be homozygous, where homo means same. And if the variants carried by the animal are different, then that animal is said to be heterozygous, hetero meaning different. The other word that you should know is phenotype. And a phenotype is a trait, basically, of the animal. It's something that we can see or something that we can measure. So it might be a coat color, it might be a hip score, it might be a blood, you know, some blood parameter that we've had in a blood test. Many different things can be phenotypes. It could be a temperament test, for example. But it's usually something that we care about in our breeding program. Okay, so I was lucky enough to be a part of the group that mapped the dog genome back in 2005. And actually the Dalmatian in this picture belonged to one of my friends who's at the University of California, Davis. Her name was Mika. And the genome, the dog genome was what's called a reference genome. So that's what I was saying before, where we want a map so that when we're talking about a gene or a place, geneticists are all talking about the same gene in the same place, that we can go directly to that spot and know what's there. And so, so the dog reference genome was a really, really important advance in canine genetics and genomics. And my role in that was to discover all of the genetic markers in the dog genome at that time. And I'll talk more about that in a moment. But if you wanted to, unlike showing it as the chromosomes that we saw before, in a reference genome, we just take 1 chromosome out of each pair. So rather than showing them as 2 chromosomes, we just choose 1 and we show it as that. And this is a representation of the chromosomes going from chromosome 1 through to the X chromosome, and just showing that in general in the dog, they've arranged them according to their length. So chromosome 1 is the longest one, and chromosome 38 is the shortest one. There's a little bit of wobbling about in there because they didn't get it quite right when they named them. And the X chromosome is the long one at the bottom. So this is a girl dog, and this just happened to be a convenient shot that I had because we're doing some of you may know, we did a study last year where we were seeing how much dingo was in kelpies using just coat colour and ear loci, and we saw that there wasn't much. Well, this year we're looking more carefully, and that's pretty much how much. The red represents how much dingo is in a kelpie, and if I showed you a bull terrier, it would be a lot more red. So there's not a lot of dingo in a kelpie that we can tell. Sneak prep. So 2.5 billion, that's one with 9 zeros of those letters A, C, T, and G. And we can't possibly look at it by eye. We need computers, supercomputers. And that's why I'm the professor of computational genomics, because I use computers to look at the DNA. But one thing a lot of you may not realize, and it's kind of important, I think, when you're thinking about the COI, the coefficient of inbreeding debate, one thing that not many people realize is how little of the genome that the genes make up. Actually, they make up less than 3% of the DNA that's in the genome. So, and actually the other thing that I think is interesting in that coefficient of inbreeding debate is that evolution tries extraordinarily hard to not change the DNA in the genes themselves. So if you look at the DNA in the genes of a dog, in the actual parts that code for the protein, and you compare them with our DNA, what you find is that they're 90% the same. Whereas if you look at another part of the DNA that doesn't code for a protein coding part of the gene, they're not really alike at all. So evolution has tried very, very, very hard to keep these protein coding genes as they should be. But there's a lot of things that go on around the genes that are the little switches and dials that make them go faster or slower or tell the DNA which tissue to use them in. And so it is important that all of those things are right, or else the animal isn't made correctly. So given that we've got these 2.5 billion letters of DNA, how do we work with them? And so in the pre-reference genome era, people used to use microsatellites, and there are about 1,500 of those that have been published for the dog. And that's like one marker. So these are basically all genetic markers, and that's one marker for every 1.6 million letters of DNA. So it's quite a big gap it's a very, very sparse picture of what's going on in the DNA. One topical thing is that there are certain products that are being sort of promoted as being ways of assessing diversity in dogs, one of which is, for example, the better bred system, but there are others, it's not the only one. And a lot of these systems use a single region of the DNA, which is called the major histocompatibility complex, to assess the level of diversity in a breed. And that region is very small. It's about probably only about 2% of the DNA of the dog. And so I personally think that if you want to assess diversity in the genome, there are better ways of doing it than looking at a 2%, a very small section of the genome. The dog is much more than its MHC. Okay, so in the old days for dog parentage analyses, they used to use a couple of microsatellites to assess dog parentage. That has changed now. Now they're using a different kind of marker. So the next kind of marker that we work with is called a SNP, single nucleotide polymorphism, or a SNP. So here we have tools that have been developed. These are actual structural constructs. These are actual a glass slide with actually DNA embedded on it. And so it works to capture a particular known variant at a particular known address in the dog genome. You can't just put stuff down and hope that you're going to find a variant. You need to know first where that variant is. And so most of the products that are used routinely for assessing SNPs in the dog genome have been designed. And the first construct, the first arrays that were made for the dog genome, I actually designed a couple of them, and they used the markers that we had discovered when we did the dog reference genome. So we're not ever going to see anything new by this method. We're only ever going to see things that we already know about. But what it can do is it can help us to find areas that are of interest to us. And that's what I do for, what most of my dog work that I do is gene mapping. And we use these tools to help us to find genes that are responsible for traits that are of interest to people. So now one thing is that these products are fairly flexible, and that if important new variants are discovered, then they can be added into these genome scans. And these are the kinds of products that are used by Orovet, My Dog DNA, Embark. All of those people are using SNP scans to do their assessment of your dog's disease status and diversity and other things that they like to do with them. Now, these products are limited in various ways. One way that they're limited, depending on the product, is that they can be quite sensitive to the quality of the DNA that's provided to them. And so I think one of the questions that we had was asking about using scans for doing disease testing in dogs. And I will tell you that I can purchase wholesale DNA scans. And so I do get all of my dog's DNA scanned using this kind of strategy. But if I want to report a genetic test result for my dog, what I do is I purchase the laboratory test for that single variant from an accredited laboratory, and I send a good quality sample to do that. Because if you are using a cheek swab and you're asking the laboratory to read 230,000 DNA markers from a cheek swab, and in my experience, owners are not particularly good at collecting their own cheek swabs. They generally don't. They have a quick swish. They think it's CSI, whereas actually you have to be in there for quite a long time to get enough DNA to do anything of the order of an array with. What happens then is the software. So what happens is that DNA adheres to the probe on the slide, and if it matches, it lights up, and if it doesn't match, it doesn't light up. So if you put on that slide bad quality DNA, by which I mean you might have a very, very low concentration of DNA, then you're making that little bit of DNA try and go a long, long way. And what happens is the signal is very, very faint. And so what can happen is, if you have a the way the array calls is it has a probe for the two possibilities that it's looking for. Does it have this one or does it have the other one? And so if it has the normal one, it would light up and it would say, that's the A signal. Yes, it's lit up. It's got a nice big A signal. And then it will read the other one and it will say, it will look for the alternate, which might be the disease allele. And it will say, okay, we see nothing for the disease allele, therefore the dog is normal. Okay, so now you have a situation where the dog has a recessive disease, in which case the signal for the B allele is high and the signal for the A allele is low. And so ideally, if the dog is a carrier, then what will happen is they'll both light up and you'll get a bit of signal for both of the markers. Now, if you're putting low quality DNA onto that slide, what happens is you get a very, very faint signal from both. And so what the software does, if it's not calibrated properly, is it says, hello, they're both the same size, therefore the dog is a carrier. And so that's why I don't want to put a cheek swab onto an array platform, because I want my dogs, I want to have one test done with my cheek swab so that I don't have to spread my DNA across 230,000 markers. Instead, I'm just going to put it on one marker, and then I know I'm going to get a good quality result because there'll be plenty of DNA to look at that one mark. All right, so that's sort of a little bit of practical usage there. That I will say though, I tend to use these as a heads up test. You can get a pretty good idea. I have had one scan that I did once that came back with an incorrect result, and the accuracy on them is about 98% when you have good quality DNA, but it falls down to about 90% if you have poor quality DNA. So that means one reading in every 10 could be wrong. So just be aware of that. Another question is, how soon can you test? I think it's no point doing it unless you can individually identify your pups. So in toddlers, it's usually fairly easy because they have white markings, so you can usually tell them apart. But if you had black Labradors, it might be a little bit more challenging. But if you can individually identify the dog and individually connect that swab, unequivocally with that puppy, so that later on you'll get the same puppy with the same result, you can do it very early on. And in fact, we did a research publication where we actually squished umbilical cord onto the testing medium, and that worked fine. So you can do it at birth if you want to. Like I was just collecting placentas and just taking the placenta side and just squishing it onto the, in our case, we used FTA cards, but you can use a swab. You could probably use, I don't know, if it shouldn't be any different than a cheek swab. And it did all come back as true to the puppy and not of the mother, as long as you do it on the actual cord itself. Otherwise, you can do a cheek swab, but you need to have the puppy when it hasn't been on mum very recently. All right, so we use these SNPs to map things. And then when we get to the place that we want, the gold standard of knowing what's in your dog's DNA is a whole genome sequence. And in that case, we read about 95% of those 2.5 billion letters. And what happens is that when we do that technology, we read each place about 30 times. And that allows us to discover actually new variants that might be related to traits of interest. But it's considerably more expensive, although not so expensive. When I was involved in the horse genome sequencing project, and it cost $12 million to sequence the horse, but now you can do the same thing for about $1,000, so it's not too bad. However, in order to then interpret the results, you need software, you need computers, you need scientists who know what they're doing. And so these are research metrics typically, rather than things that an individual dog breeder would use. Although we have proposed that if you have sires that are used way too many times, it might be worth having them sequenced so we can predict anything coming down the pipeline that might not be very good, to help for genetic testing in the future. That's kind of a researcher viewpoint on things. Now we get back to things that we're a little bit more familiar with. And so our Mendelian inheritance, so these are the traits that are simply inherited. And so with recessive inheritance, what that means is you need to get a copy from mum and dad in order for that trait to appear. So there's no point with a recessive disorder blaming dad or blaming mum. It's both of the parents that have passed on that trait. So in my family, the red hair, my dad had red hair. I don't have red hair, and neither do any of my brothers and sisters. However, nearly all of my brothers, and I don't have kids, but all of my brothers and sisters all have red-haired children, which possibly speaks to the fact that 2 of them married redheads, but also it shows that we all carry that red hair gene, and so it's come out in the next generation. So my mum probably, I won't say definitely, but very likely my mother did not carry red hair, and that's why none of us children have it. All right, so if you have 2 normal individuals, genetically normal individuals bred together, all of their progeny will be normal. If you breed a normal with a carrier, you get 50% clear and 50% carrier. Now, this is presuming that the carriers are phenotypically normal, which means they don't have a disease. If you breed a clear with an affected, you get all carriers, but these dogs are all normal. And so it always irritates me when everybody's trying to get rid of, you know, you can breed an affected dog safely, actually, for a recessive disorder, but you need to be aware that all of the progeny will be carriers. And so if you don't want to do the genetic testing, you want to move towards everybody being clear. But if you don't mind managing things with a genetic test, then it's okay to breed. As long as it's a purely Mendelian recessive disease, it's actually okay to breed from an affected dog, as long as you understand that all of those progeny will be carriers and then all have the possibility of throwing that disorder if they're bred with another carrier. So the carrier by carrier is your traditional Punnett square, where you get 50% carrier, 25% clear, and 25% affected. And if you breed a carrier with an affected one, then it's the opposite ratio from up here, where you get 50% carrier and 50% affected, which is a situation we don't really want. So this would be like, I can't think of a good example there, but it's not something you really want to see. And obviously you don't want to breed affected together. In the old days, people used to do test matings to see how things would turn out. And I'm not a proponent of that at all. I don't think in this day and age that we can really justify that from an animal welfare perspective. You're not ever going to clean something out with one breeding. So the best trick is, if you have something that's clearly a recessively inherited disorder in your breed, it's better to make a test for it. Okay, so the next one, so in this case, even though these dogs are all different colors, when we look at them, we'd only see 2 phenotypes. We would see normal, which are the clear dogs and the carrier dogs, and we would see affected. So this might be your PRA, would be like this, where you have an affected dog. And another question that someone asked was, they had a PRA, a dog that tested as affected for PRA, but did not appear to be blind. What did I think of that? And I would say that they don't all go blind at the same age. It may be quite old before it goes blind, or it's possible the test result is wrong. And so if you want to check, I would go and get a single test result from a lab that does single tests as opposed to a panel test. All right, so 2 phenotypes there. The next one is, in my breed, we have a gene called CDDY, which stands for chondrodystrophy. And what it does is it does a couple of things, but the main thing that people would see that it does is it changes the conformation of the dog. And it tends to, most of the, I've got little ribbons on the blue ones here, because it turns out that in the show ring, nearly every champion show dog is a heterozygote for this gene. When it's homozygous, the dogs are very short in the leg, but they have very good angulation. But the other thing is that in this particular case, both of the animals that carry this CDDY have a higher risk of having back problems in their life. Now, given that this gene is in such a high frequency in the breeding dogs, most litters would have this wild type produced, and you'd ask, well, why don't people want the wild type? And the reason is that they're longer in the leg, they're a bit too tall and fine, and they tend to be a bit straighter in the shoulder. So the breeders really like this one. Some of them really like this one. Personally, I really like this one. You can get good conformation in this one, but it's hard to find. So what ends up happening is that they all trend towards the middle here. So it ends up being very, very common to have a carrier dog in your breeding dog. A question was just asked, what if there is no single gene test in your breed? Then the answer to that is you collaborate with a geneticist to collect enough samples to make a test. Because honestly, that Schnauzer test that we made, we had 3 affected animals and we made a test for them. So it takes a bit of money, not too much. I think that test cost about $5,000, but it's about people power. So you need a student who's interested to do it. You need a lab that's interested to do it. You need at least 3, possibly more case samples. And you need people who are willing to share the information, because honestly, that's the big limiting factor. Last of all, we have dominant inheritance, and I've used the little rosettes here just to designate the carriers. The heterozygous ones are not any better than the black ones. This is our traditional E coat color locus, and it's just the same. These are all, you'll see that actually each of these pictures is exactly the same. It's just that whether you can see the carriers or not. In this case, you can see the carriers, but in this case, in the recessive and the dominant case, you can't see the carriers. Whereas in the incomplete dominant or the co-dominant case, you can, the actual middle ones look different. Sorry, there was a chat there. Oh, how do I see it? Chat. Okay, everyone saw that, sorry. All right, so someone was asking about deafness. That's a complex trait. That's why there's not a deafness gene. There are many deafness genes. All righty, so next. Whoops, come on. What's happening? I'm scared to click the mouse because last time it took me off on a journey I didn't want to go on. All righty. So next, the next thing that a lot of people ask about is relationship and inbreeding. So as I've already alluded to, each of those chromosomes that comes in a pair, except for the X and Y, which are their own thing, you get one in the progeny comes from dad and one comes from mum. But the one that you get from dad isn't a whole one. It's a mixed up version of the one that dad got from his mum and his dad. So here I've depicted that as a colour smushing. So that mum's one's kind of mauvey coloured and dad's one's kind of limey coloured, because it's a blend of his 2 dad ones and that's a blend of her 2 mum ones. And so in the progeny, it's not any one of these, it's a combination of all of them. And so there's a shuffling that occurs, and this is how we get genetic variation in species. One of the things that leads to genetic variation in species. Alrighty, I love this picture. I used to say these look like tollers. That's before we had the CDDY gene, and it actually is pretty close to the truth, except it's a different gene actually that causes it. This is the chondrodysplasia gene, which is on chromosome 18. Anyway, but what I want to show people, and this is something that a lot of people don't get. So here what we're looking at is a traditional plant breeding scenario in a way, where we have 2 different breeds at the top that are mated together and they look quite different to one another. And when you breed them together, what happens is all of the dominant genes are dominant and all the recessive genes don't show up. So you end up with very, very consistent looking puppies. So the next level down, so here's mum and here's dad, presuming he's got the penis. And then here and the next level down, we've got these little brown dogs, which look very similar to one another. And so a lot of people think that if you breed two dogs together that look alike, then you're going to get dogs that look just like they do. And that doesn't always happen, because when you breed these two dogs together, what you get are all of these different types down here. And the reason for that is that these two, like me and the red hair, all of these dogs carry one gene from each parent. And so this one's got, you know, one chromosome from one mum and one chromosome from dad. And this gene's got one chromosome from mum and one chromosome from dad. But in certain genes, these ones might have had two mum leg length genes. And these ones might have had, you know, one mum and one dad leg length genes. So they've sort of got the intermediate one. And these guys got two dad's leg length gene. And you can see all kinds of things going on here. You've got long ears, short ears, white markings, easty-westy legs, all sorts of things going on there. And we call this back segregation to the parental type. So what this means is that somewhere down here, about 25% of the time, you'll get a gene from mum that goes with a gene from mum, whereas here that never happens. And you'll get, or you might get a gene from dad that goes with a gene from dad. And so technically, each grandparent contributes 25% to the grand progeny. But in this case, because it's an inbred pedigree, each grandparent counts for two. Just want to check the chat. Yes, that's true. So each, so the question was that each pup can get different aspects of mum. So there may still be a bit of variation, but in general they look, so for example, the sabling is a little bit different on one than the other. So if the parent's heterozygous for something, then the puppies can be different. But if there are certain fixed traits in the breeds, then very likely you'll get this nice even effect. Now they use this in the beef cattle industry to cross Hereford and Angus to make black baldy cattle. And they grow well, you know, you get the hybrid vigor thing going on. They all look the same. They're all the same size. You can use the same cattle yards. They're easy to send to market because they all look the same. But if you breed them, and this is also the same as your Labradoodle, right? They all look kind of the same, you know, fairly predictable. But if you breed a Labradoodle with a Labradoodle, then all of a sudden all this stuff starts happening and you get the incorrect coats and you get big and small and goodness knows what goes on. So yeah, just, I guess the thing to drive home here is just because you breed two animals together that look alike does not mean that you'll get puppies that look like the parents. Oh, why am I taking so long? Hurry up. All right. Alrighty, so I'm just using my dog Burn as an example here. I see from time to time on the dog, you know, the pedigree dog chat, you know, Facebook pages and so forth, people have this idea that certain dogs are more important than others. And so often you'll hear that the sire line's important, or that in thoroughbred breeding, they think the dam line's important. The reality is that Harbour Light Salty Dog is so Edlin's second chance at Roslyn. Edlin's second chance at Roslyn has exactly the same contribution as Westerly's Superstar at Bediah, this one. and they have exactly the same as Harbour Light Salty Dog, except for Harbour Light Salty Dog is contributing the Y chromosome to this dog, and Edlin's Second Chance at Roslyn is contributing the mitochondria to this dog. How important is that? Not very important, because the Y chromosome is tiny. And on the mitochondria, even though the mitochondria are the powerhouse of our bodies, they have about 1,500 genes contribute to the mitochondria. But what you may not know is that most of those genes that make the mitochondria actually live on the non-mitochondrial genome. So they live on these things here that we call autosomes, which is where we've paired up the 38 chromosomes with each other. So nearly all of the DNA that's worth anything is on these 38 pairs of chromosomes that are inherited equally from both sexes. The Y chromosome is mainly concerned with making sperm. Both the X and the Y chromosome have a lot of repetitive content, which is very difficult to work out what it does. There are some important genes on them, but, and as someone else was asking about X-linked inheritance, so for example, hemophilia is on the X chromosome, and what that means is if you're a male, you always get your X chromosome from your mum, and so you only have one. So if mum carries something on her X chromosome and you happen to, mum's got two to give away, but if you happen to get the one that's bad and you're a boy, there's nothing to match it, so it acts as though it's homozygous. If you're a girl, you've got to get two bad ones from mum. Oh, sorry, you've got to get a bad one from mum and a bad one from dad to have the effect of that hemophilia. Whereas if you're a boy and your mum carries hemophilia, you've got a 50-50 chance of being hemophiliac. If you're a girl and your mum carries hemophilia, it really depends how common it is in the general population and how related she is to dad to determine your chances of being affected. Okay, so the coefficient of relationship tells us what proportion of our DNA is shared between us and our relatives. So for example, I share 50% of my DNA with my mum because I got one of her chromosomes from each pair from her, and I share 50% of my DNA with my dad. And on average, but not all the time, I share around about 25% with each of my grandparents, not all the time. Theoretically 25%, in reality can be anything from 0 to 50%. Oh, I did the wrong one. So back to this slide again, just to remind us that we get one chromosome from mum and one chromosome from dad, and they're a shuffled version of the grandparents. And so then each generation step, so this is like a generation step, each generation step diminishes the relationship by a half. So you need to know your half times tables, which for those of us who work in computing, it's not that hard because it's kind of similar to working out, you know, how much power your computer has. It's the binary sort of system that they use is the same. All right, I thought I'd talk a little bit about parentage profile versus parentage verification. And so having a parentage profile done doesn't really tell you very much. It's just like getting a library of markers that can then be used to verify pedigree accuracy if required. Whereas parentage verification requires at least 3 animals to be tested. Those would be the puppy and the presumed parents need to be tested using the same markers, because we need to check that the puppy doesn't have markers that didn't come from either of the presumed parents. So I've got here, sorry. Okay, so we'll get to that. All right, so I've borrowed this from a cow website because it just showed each little squiggly line here is kind of some markers. And in each case, the offspring is the top line, and then the mother and the father are the next two lines down. And so what you want is every time there's a little lump here, you want there to be a little lump down here. So in this case, that lump, there's one there. That lump, they both have it. That lump, mum has it. That lump, nobody, it got something. It didn't get that one from anybody. All right, so these parents might have been carriers for that lump or something. So in this case, the progeny doesn't have anything that isn't seen in the parents, so that's okay. But down here, we have this pesky little lump here, which is neither in mum nor in dad. So that's a pedigree verification fail, that one. Now, I don't do this myself, so I'm kind of, I know roughly how it works, but I don't do it at all in my lab. So we can use SNP markers to do something similar, but you need more of them. And these days, actually, the parentage profile has all gone to SNP markers. So that's what we were talking about before. All right, so there are commercial products called DNA profiles. And in these, you can purchase a parentage profile. So not parentage verification, it's just a parentage profile. Coat color markers, common disorder mutations, where it's been made possible on that platform and when it's publicly available. But you can miss some of the emerging disease tests for each breed. And so I have an example here. Go to the Tollers. So this is the Orovet profile for the Tollers. I got a little bit distracted when I found this because it shows here one of the traits it's testing for is black hair follicular dysplasia. And this stopped me in my tracks because, first of all, the variant for that disease, that's a disease, the variant for that disease is not published. And Tollers typically don't have black hair. So I'm not quite sure why that one's in there. But it gives you the ISAG, the International Society of Animal Genetics Canine 288 SNP panel. And I downloaded the ISAG 288 SNP panel. But unfortunately, I also had a look at a sample dog profile from the system online. But unfortunately, I was unable to reconcile the markers that were reported here with that SNP panel. So they may be the SNP panel markers, but they don't have the same name, which means that they may as well be entirely different from the point of view of someone wanting to use them again. So I think that if the ANKC wants to have people use this panel, they need to have the markers be clearly identified according to the ISAG names, because otherwise those markers cannot be used by a different company. Got a chat thing up here. Okay, alrighty. So that's my breed. Now, in my breed, this parentage profile is offering up cleft lip and palate, which is actually a disorder test that I was involved with creating, collie eye anomaly, degenerative myelopathy, which is in most breeds, which I was also involved in making that test, PRA, PRCD, which is definitely something we want, and then a few coat color traits, some of which are more important in our breed than others. So your breed will look like that, I'm guessing. Well, not like a Toller, but you know what I mean. There'll be some tests that look good for you, and there'll be some tests that don't look good for you. And I'll talk in a little bit about how you can find out a little bit more about which ones you should be using. All right, so next, the coefficient of inbreeding. So we talked about relationship and how you tell, how related animals are and how each generation step that relationship diminishes by half. Well, the coefficient of inbreeding is telling you, on average, how often do you expect this puppy to have exactly the same piece of DNA inherited from mum at a gene as it got from dad, and by the exact piece of DNA, I mean exactly the same piece of DNA, because they came from the same ancestor in mum and dad. So it's only possible if mum and dad are relatives to one another. And by definition, it has to be homozygous at that spot. But it also describes the average effect of that across the whole individual. So by definition, a coefficient of inbreeding in any pup is 1/2 of the relationship between the pup's parents. So if the parents are not at all hardly related, then the pup's not likely to be very inbred. So what's the difference between line breeding and inbreeding? Well, this will get everyone going, I'm sure. But, you know, from a geneticist perspective, there's no difference because they both involve breeding related animals together. And it's just, you know, you might have your special formula for how you do the mating so that it benefits your breeding program. But from a geneticist perspective, they're just different shades of the same color. They're both the same process. You're breeding relatives together. And so there's a chance that in their offspring, the progeny will inherit a piece of DNA from mum that's exactly the same as the piece of DNA that it inherited from dad. And if you were to breed an animal with itself, though, it might surprise you to learn that the inbreeding coefficient of the progeny is only 1/2. So even if that animal bred with itself, which is not physically possible, the relationship coefficient, the inbreeding coefficient of the progeny is only 1/2. Now, I know lots of people get fired up about inbreeding. So how close is too close? And I just got here, you know, there's a lot of debate about this online. There's a lot of debate in the genetics community as well. And to me, it's not all the same. So to a lot of mathematical population geneticists, they would regard these two COI things as the same. But to me, they're not the same, because to me, and my understanding of genomics and the way DNA is passed through generations, the first case is nearly not inbred at all in my mind, whereas the second case is quite seriously inbred, because this is a result of a close relatives breeding together. And that means that it's not just a little teeny weeny shuffled piece of DNA that might just happen to be the same, and it might not even land on a gene, because remember, the genes are only 3%. What's going to happen in the second case is you're going to get like 1/2 of a chromosome that's the same between mum and dad. So that every little switch and dial that turns the genes on and off, and every gene is going to be the same between mum that the puppy got from mum and dad. So the question is asked, could this be regarded as an arbitrary measurement and not important on a genomic scale? It's difficult to say. I mean, it's one of those things where it all relates to risk. It's like you can't, every risk is a risk. And so my sense is that as long as people are selecting to breed from healthy animals, the risk of this long ago inbreeding is fairly small. If you have people that are breeding from unhealthy animals, then the risk of that long ago inbreeding is a bit higher. Okay, so in my view though, personally, I would much rather see the first situation in a pedigree than the second one in my own. And if I want to give people advice, I'd say, apply the same rules as we use for humans. You know, you don't marry your dad. It's probably a good plan not to breed with your brother. And a lot of people say that's anthropomorphizing dogs, but I would just say that's biology, and biology says that we shouldn't do that. So the reason those rules have been made for humans is because it doesn't work out very well. The other rule of thumb I would say is to try and breed so that in your pedigree, and maybe I can back up a little bit to Burns' pedigree. Let me see how I go. No, it's not going to work. Anyway, in your pedigree, you've got mum at the top. Oh, sorry, dad at the top and mum at the bottom. And then you've got all mum's ancestors, sorry, dad's ancestors and all mum's ancestors. If you draw a line between the two, what you want to minimize is how many animals are in mum's pedigree and in dad's pedigree, because that means that mum and dad are related. And so I typically, you know, someone said to me once, and I think that's pretty good advice, is if you can't see the relationship on a standard pedigree, don't worry about it too much, because I think that there's way too much angst that goes into COIs, that much more than they deserve. People bred without, you know, 10 generation COIs for many, many years. But you know, I'm not a fan of close breeding, but the 10 generation COI is probably more angst than is required. All right, so those are what my suggestions are, and that's not always possible either. But you know, just try and minimize the relationship between mum and dad in the pedigree. If you can minimize the relationship between mum and the dad in the pedigree, then you're going to avoid the worst of the things for which there is no test. If there's a test, you know, there's always things, let me say there are always things for which there is no test. There are always things for which there is no test, because new disorders emerge in all the time. We've dealt with many of them in my laboratory that have recently emerged genetic disorders. All right, so each time your body makes an egg or a sperm, there are mistakes in it. And if those mistakes happen to happen in a gene, and if that puppy goes on to be an extremely popular sire, then those mistakes can be spread far and wide. Okay, so from a genetics perspective, inbreeding is breeding together the related individuals. Geneticists, everyone else does, but geneticists don't differentiate inbreeding and line breeding, because they both involve breeding related individuals together. However, in my discussions with people over the years, what I think I have come to realize is that people often, a lot of people say, I use line breeding, but then when I look at the breedings they're doing, they're probably not a lot more closely bred than something I would do. And what they mean is that they're selecting for particular traits in particular lines of dogs. And that's not line breeding, that's selection. And if we're not doing selection, we're not doing our business right, in my view. So selection means you're concentrating desirable genes in the puppies. And typically the way you do that is you breed together animals that have those desirable genes, and sometimes those animals that have desirable genes will be related. However, if you are breeding relatives together just because they're relatives, then I would suggest that that's a very unhelpful thing to do. There's no benefit, there's no magical benefit of line breeding. There is no magical benefit of line breeding that is above and beyond what's possible from selecting for those same characteristics in less related individuals. And you'll have a lot healthier pups and bigger litters and so forth if you go that way. And I guess there's a lot of research that's shown that for each 10% of COI, you lose about a puppy a litter. So it's not worth it. There's so many things that we don't have tests for. The very, very vast majority of things we don't have tests for. And like I said, there are diseases that emerge every generation, and it's just a question of how dispersed they are in the population as to whether you see them show up again or not. All right, so Margaret said at the beginning that I'm on the expert panel for Dog WellNet. And there are a few questions relating to how do I know what tests are for my breed? How do I know which tests are good or which ones are bad, which ones I should be using, and so on and so forth. So this Dog WellNet is an international organization that grew out of the different kennel clubs internationally. It was started by the Swedish Kennel Club, and there are many different kennel clubs around the world that have contributed to this. The genetic test providers also contributed to it, unfortunately not enough in my opinion. But the whole thing, because it's, you know, kennel clubs are typically volunteer-based organizations, Dog WellNet consists of 1.5 people who pull all this information together and harass people like me to give content and do analysis and stuff like that for free. And it struggles to stay alive. And so I would be encouraging ANKC to get on board and support Dog WellNet. And I know that ANKC always comes back and says, well, the member organizations have to do it because we don't have any money and so forth. But I would urge people to get behind this, because it's non-partisan in that the advice they give is untainted by commerce. And that's the reason they have panels of experts like me on there, is to assess the quality of the information. All right, so on Dog WellNet, so dogwellnet.com, if you go in there, the front page rotates, so it may not be this picture, it might be a different one. And if you go in there, oopsie, sorry, didn't do that very well. All right, so if you go in there, I don't know why they chose this name, but it's called the Harmonization of Genetic Testing, which I think must be some Swedish thing, because I've never heard anyone in an English-speaking country call it that. But basically it's talking about, it's trying to get across the concept of using genetic tests wisely for the benefit of breed health and welfare of purebred dogs. That's what this site's all about. And so if you want to learn more about your breed and what can go on in there, and there's a whole lot of different resources on here as well. But in terms of just like, for example, the genetic testing part, what tests should I be using in my breed? Where can I get them from? Which ones are good and which ones should I ignore? This is a good place to go. So if we select that orange box here, it's not an orange box on the thing I put the orange box on, then it brings you, you might have to scroll down a bit, but it gives you to this Harmonization of Genetic Testing in Dogs HGTD quality testing database, where you can search by breed. So then if I type in Nova Scotia Duck-Tailing Retriever, then it gives me the tests that are offered in my breed. Oops. So it says here, coat color dilution alopecia, which in our breed is really just dilution because we don't get the alopecia part of it. So, and here it says, you know, chow chow, but it's just coat color dilution. It's a well-known test. It's valid in our breed, and we call it buff. Then we have chondrodysplasia, which is not the one I showed. This is the Basset Hound version. That one's offered in our breed, but it's not relevant to our breed. So it gets 1 red paw, meaning don't bother. Then we have the chondrodystrophy type 1, IVDD. That was researched in our breed, so that gets 4 paws, 4 green paws. That's a good one if you want to use that. Collie eye anomaly has been offered in our breed for many years, but recent research shows that it doesn't actually do anything very much, so that's been given a red paw. And degenerative myelopathy has been seen in our breed. It is known to have a clinical effect in our breed, and so it gets 4 paws. But it's one of those complex disorders, and I know that's going to get everyone going because it's in every breed. And it's not equally relevant to every breed. So in our breed, it's not fully, it has a thing called incomplete penetrance, which means that even if you have 2 copies of the DNA that says you should get it, you may never have it in your lifetime. So, and it hits dogs really old. So, you know, you have to weigh up if it's worth doing or not. For me, a lot of these things depend on how frequent they are in the breed, how devastating they are to the owners, and these are often breeder decisions. Then we have, so the genetic profile did offer this, but you had to pay extra for it, did offer this. It did not offer this one, JAD, juvenile Addison's disease, which is a big one in our breed. but it's only offered through one lab and has not been published yet. So it's not generally available to the Orovets, My Dog DNA is an Embarks of the world. You have to actually go to the research lab group to get it. Same goes for this other one here, degenerative encephalopathy. It is in our breed. It is true for our breed, but it's not available on the panel tests. And then cleft lip and palate is true to our breed, developed in our breed. Whether you use it or not, it's up to you, because it's puppies, and people have ideas about breeder things versus puppy owner things. PRA, I'm not sure why it has 3 orange or yellow paws there. There's some sort of warning involved with that. I think it's fine for our breed, but your breed will look like this. So there will be experts that are offering opinion on whether these tests are of value in your breed or not. Not all the breeds are done because the expert panel doesn't know about all the breeds, but it's probably the best resource out there, and it's being improved all the time, and there's a big group of us working on it. So this is what we're doing, is to try and sort the wheat from the chaff in terms of the tests that are being offered for different breeds. And this is where you'll find the most up-to-date information. So that's where I wanted to get up to in terms of the general presentation. I do have more information available on coat colours or estimated breeding values if people want to go into that. But I think what we should do is stop the sort of lecture part there and just go back to answering your questions and discussing the things that are of importance to you, and perhaps Margaret can take over and control which questions that I answer for you. So thank you for that. You're all very quiet. It's kind of weird in a way. I'm used to students asking questions, and we did have a few questions on the way through, so thank you for those. I don't know if everyone saw them the way I did, but no, that was good. So I'll stop my screen share for now, and we can just go over to general questions.
Thanks, Claire. There was actually one question there that, I just wanted to put in an extra plug and say that my concept was that DogWellNet is like having choice for dog breeders, the Australian choice option, that it's a way to review what's being put on offer for us.
Like the magazine choice.
Yeah, like the magazine choice.
Yeah.
So back here we had a question that's, and I'll just read it word for word. If a dog appears more than 4 times in a pedigree within the first 3 generations, how much influence would that dog have on puppies?
Depends if it's on one side of the pedigree or both sides of the pedigree. So by one side, I mean on just dad's side, or if it's on both dad's side and mum's side. The reality is that short of cloning, it's not possible to recreate a dog by line breeding. I know people think that they can, but you can't, because...
Oh, the answer to that is on both sides of the pedigree.
Yeah, so I'd say that's a very not ideal. Oh, so this is another thing, right? If you have, on one of the forums I was on, someone was asking, should they have the Embark dog diversity test done on their dog? And they would figure out how inbred their dog is, because they had worked out that in a litter of puppies, they may not all be inbred the same. And that's true. But the reality is that once the puppy's on the ground, it's done. You have what you have. And how inbred they are is kind of irrelevant. What is relevant is how related they are to their mate if you breed them. And in that case, this case we're talking about, we have a dog multiple times on both sides of the pedigree. What's happening there?
I don't know, either you or I have shared our screen.
No, it's not me, I don't think.
Ah, must be me.
So when you have a dog, I guess the most inbred dog I've ever seen in my life is a crossbred dog. And I think this is a very common misconception that I try and clear up whenever I'm able to. in the top, Margaret, you have to pull the thingy down and then go stop screen share.
Yeah. Oh, you've got to pull it down. Oh, right.
Stop.
Yep. You're viewing. No, it's somebody else's screen share. Could Clara Kennels remove? I don't know. Somebody's.
You should be able to stop it for them, Margaret.
Yes, I should. You're right. I've suddenly lost all my member view options.
So we're viewing Clara Kennel's screen.
Chops. There we go.
Okay. So where was I? Oh, I was going to say a very common misconception in the public, and this relates to particularly oodles, I guess, is that if a dog is crossbred, that it can't be inbred, and that is so not true. You can have an oodle mated with its brother or sister or father or mother, and the progeny of that will be more inbred than your average pedigreed dog. Now, as I was saying, the very most inbred dog I've ever observed in my life was actually a crossbred dog. It's a livestock herding dog, and it had the same ancestor 19 times. That's certainly, that's a lot of that dog. And they were obviously trying to recreate that ancestor. But as I was saying, when you line breed, the result is that you get an animal that's very homozygous throughout its genome. The dog or bitch that you're trying to emulate in your breeding program by using line breeding wasn't like that. That dog was probably majority heterozygous. And so when you're line breeding, you're never going to achieve the same heterozygosity of the same genes that was in that originating dog. It's not possible. So unless that dog itself was so inbred, the only way you can replicate it is by cloning. So to use line breeding to emulate an ancestor doesn't work. Sorry, doesn't work. You can use it for creating consistency, but it doesn't work for recreating an ancestor. Margaret?
Now, are you, thank you for that. Now, are you best to head across to the Slido questions, or have you addressed most of those through your points that you've raised within your presentation?
There are a few others. Some of these I've not got. Let me just try and pick up. I should do the Slido ones, and we'll come back to the chat ones because the Slido ones were there first.
Yes. Yes.
Alrighty. Let me have a look. Uh-oh. Here we go. Alrighty, let me have a look. Have people been liking things? No?
Oh.
Okay. So other than word of mouth, is there a way to evaluate which lab is better to test through for your particular breed, and does it even matter what breed? I think Dog WellNet is one way to know. No, Dog WellNet is not in a position to say this lab is good or this lab is bad, because those labs are giving them money, and if they don't, they're not there to say one is good or one is bad. I think you need to understand that different labs are good at different things. So as I was saying, the genome scan ones, they're cheap, but do I want to run it on my breeding animal to have its breeding test result that's recorded if they either recorded on the pedigree? I want to have a single gene test done for my dog so that I know that that result is right and that that lab will redo the test if there's any problem. That's what I want. I use the scans for heads up. And I actually think they're quite dangerous for breeding, really, because your dog could be labelled with a result that's incorrect for life. So, yeah, I don't know. You've got to figure out what you want to do. And I think as a community, we need to really be aware of that they're not perfect. and that there are many places the errors can creep in. It can creep in from the wrong dog being collected to the swab not being treated correctly, so it's contaminated with bacteria, to the lab misidentifying the swab, to the test having deficiencies in the reagents so that it doesn't work properly, to the test in that breed not working because there's another genetic variant that happens under the bit that it sticks to, so it doesn't stick properly. There's many different ways that things cannot be perfect. The best way is to have a single gene test that's known to work in your breed. So that's, in terms of evaluating labs, for my breeding program, I need to go to at least 2, maybe 3 different labs to get together the tests that I want for my dogs. There really isn't a one-stop shop yet. And when people are doing commercial in confidence testing, that doesn't help. So we tend to, in our group, we publish. I don't sell tests, just FYI. I make tests and other people sell the tests that I make. And I discovered the degenerative myelopathy variant, and I have never earned $1 from it yet. Maybe one day I will, but not yet. Haven't seen a single dollar from it. Anyway. So
just to be clear, Claire, let's use my breed as a particular example. So it has a PLN gene issue that is affected by 2 variants that was developed by the Penn lab in the US, right?
Yeah.
And then it also has a microphthalmia issue that was developed by the Finnish people, complex inheritance. So I'm better off to have those 2 tests tested at 2 different labs, the 2 different labs.
I would say so, yes.
Yeah, right.
Generally, if you go to the lab that's supported by the people who created the test, that's usually the safest option.
Yeah, even though they're being offered by other laboratories.
Yeah, I think you need to be careful when other laboratories are offering them. For a heads up, it's okay, right? Because if you send, the other thing I'd be suggesting is that people make sure that the DNA sample they send for scanning is high quality. Usually that means if you've had someone collect it who knows what they're doing, or your veterinarian, blood's usually good, unless it's greyhound, because they don't have any white blood cells. Well, they have some, but not enough. Yeah, so you can get quite different. Dogs differ enormously in how much DNA you get out of the same quantity of blood. It's just astonishing. Yeah, so high quality DNA, single gene test for breeding, I would say. Yeah, and then other than that, you want a lab that's ideally got some sort of certification. ISAG is the one that we look for. The International Society of Animal Genetics is a good thing to be looking for, especially for parentage markers, because they routinely test labs for their parentage markers. Okay, so next down here, are there minimum standards on sensitivity and specificity for genetic tests to be offered by laboratories, and are they required to meet any QC requirements? The answer is no and no. No, you can offer any test you want that people are willing to buy.
So that's why you're saying you're better off to go back to the accredited laboratories.
Yeah.
And I think, yeah, I agree. I personally think that's really important. When they run tests in laboratories these days, is it just a single test, or do they split the sample and run 2 tests and compare?
It depends on the lab. I know that so for a lot of our toller tests, we use the veterinary genetics laboratory at University of California. And I know that they do all kinds of tests. They do triplicate, and they do sex and color markers to check that the animal is as it's declared to be. So they do all sorts of QC there that you may not get in other labs. So I don't know I don't know what all the labs offer. I think you just have to ask the questions and hope that they're usually, if they're been around for a while, they're usually okay. So then the next question is, are missing teeth hereditary? The answer to that is yes. Do we know the gene for it? No, not at this time. Not at this time. Then are genetic testing results affected by the time between sampling and testing or the quality of the sample? Yes, they are. Depending on the medium that the sample is collected on, it can matter or it might not. So depends too if it's a panel test or a single gene test. So usually single gene tests are a lot more robust to mistakes than panel tests are. Okay, so I breed, some breed. How do I find out the tests I need to do for my breed? Then DogWellNet is the place to go, I think. At the moment, it's the best one out there. Can off-standard coat colors be tested for, specifically black and tan, blue and tan, or brindle? I can't remember which breed this relates to, but yes, they can be. So black and tan.
Italian Greyhounds.
Italian Greyhounds, okay. So black and tan can be the A locus, but it can also be the K locus. So for example, in the Sheltie, black and tan is coded at the A locus, but in the Kelpie, black and tan is coded at the K locus. Brindle is definitely the K locus. Blue and tan is dilute, so that will appear in the test as coat color dilution alopecia, even though they probably don't. Not many breeds get the alopecia part of it.
Oh, we get it. It's fine. Sorry, I'm interrupting you because this is a really big one in our breed of dog, and we don't really have our colours completely mapped yet, as far as I can tell. So a lot of people are getting these tests done and saying that they have clear or not clear, or carriers and not carriers, which is a really big worry, in my opinion. I'm going now.
Oh, so this might be the problem I was alluding to before, where you get if your DNA quality is not there, you get all these carrier results coming out. That might be part of it. So I would say don't worry about it unless you see the color turn up in a litter. And if you see the color turn up in a litter, then you can do some targeted testing to see if it agrees with what you're seeing or not. That would be my advice there. But those are typically A locus, K locus, and melanophyllin dilute is the blue. Alopecia also in sighthounds is a different thing. There's been some work going on in that in Norway, I think. So I'm very interested in sighthounds. I do a lot of work with greyhounds actually, so these are all kind of interesting. Alrighty, so what can be done to determine what causes hip dysplasia? 2 generations of dogs have low hip and elbow scores, yet they produce progeny of hip dysplasia. Why? I guess the first thing I'd ask there is, how was it determined that the puppy had hip dysplasia? Because honestly, if it was a veterinarian doing an Ortolani sign, I would be querying whether that dog has hip dysplasia. And the other side of that is that it's a complex trait. So it's and people have historically used a bit of dodgy scoring. So, you know, if you have quality scoring, not in a way that favors your dog, but that tells you the right answer, this shouldn't happen very often. And if it is happening, the first thing I'd ask is, how was it determined the puppy had hip dysplasia? And because there's all sorts of errors that can occur that way as well. So the answer is I don't know, but there are a few things to look out for. Okay, again, validation accreditation, there isn't any. A little bit for parentage, but not much else. inbreeding coefficient. What sort of numbers are acceptable and what isn't? And is there an easy way to calculate these? It's a question of risk and how long ago. So as I was saying in my presentation, I'd much rather see a low 5-generation COI. I don't care about how many generations you go back, but try and keep it out of the recent generations would be my advice, because that's where all the problems come out. The other ones will have been selected away from, one would hope. Okay, so next one, I would like to know what genes are needed to try and get a black and tan Shih Tzu. Also, a solid red or a solid red with a black mask. Well, not knowing anything about black and tan Shih Tzus, the first thing you'd need to ask is, do they exist? If they exist, then you need to breed into families that have them. Solid red is MC1R, or red with a black mask is also MC1R. It's they're both the E locus on your, they're different variants on the E locus.
So Claire, how do we learn what are the DNA requirements for the phenotypes that we're looking for? You're sort of reeling them off so well, but where's the resource to find that out?
There's a very old book from the 1950s called, what is it called? It's by Clarence Little, and it's called Coat Color Genetics in the Dog, I think. It's from like 1950s, and it actually describes accurately most of them.
Wow.
Not all of them, because the K locus didn't exist when that was made. But it covers it pretty well for most things. All right, so also if we have a little bit of time at the end, we're getting close to time, but I do actually have some coat color slides I can quickly go through for you.
Oh yes, somebody's just saying that that book's called The Inheritance of Coat Color in the Dog by Little.
Thank you, my memory's very bad. I'm good at improvisation, but I'm bad at remembering, so that's the one. All right, so who tests that DNA tests offered by a laboratory are valid? Usually other scientists, and it usually takes a long time to work it out. And usually it relies on people complaining a bit first, and then it gets looked at. And also things like, let me just put it out there and say, if someone offers you a DNA for hip dysplasia, don't waste your money, because it's a complex trait. It's caused by hundreds of genes. There's not going to be one test that's going to give you the answer. Okay. 2 copies of a variant have been detected, for example, degenerative myelopathy, but there's very little observation of that disease in a breed. How does that happen? Well, with degenerative myelopathy, it's easy because it doesn't hit dogs till they're elderly. And so a lot of dogs, if the gene's at low frequency in the breed and it's incomplete penetrant, which means that maybe only half of the dogs, not even, maybe only 25% of the dogs that have the genetic disorder test result get the disease, and then they don't get it till they're 10 or 11, and then you're in a breed where they all die at 8 of heart disease or histiocytoma or something, then you'll never see it. So don't worry about it, because you've got other things to worry about, I would say. Yeah, so what are the susceptibility to tests offered by some companies, and how much notice of this result would you take into consideration? I would ignore them. I would ignore them for now. We've got enough to worry about. You don't need to be worrying about things that might not be anything. What is my perspective on the value of canine genetic diversity testing, such as the MBARC test and the UC Davis genetic diversity test, better bred? The better bred one is just the MHC. I have a whole theory on MHC. which is that it doesn't matter in dogs. And this is not, I don't know how to even design the experiment to test this philosophy, but my philosophy is that MHC is important if you're like a Tasmanian devil and all of the other Tasmanian devils around are just like you, then it's important to have MHC diversity. But in dogs within a breed, how do dogs live in the world? They don't live all the same breed together, unless they're beagles, maybe. But in a dog, the way dogs live is that in my house, I have 2 Tollers and a Labrador. 3 doors up, they've got a cattle dog. 2 doors over, they've got a miniature Schnauzer, and across the road, they've got a greyhound. There's so much MHC diversity there in the environment that no disease is going to wipe through and wipe out our dogs, because it's not monoculture. It matters if they live in a monoculture, it might matter, but most of us don't have a lot of dogs in a monoculture. So there's not really a lot of evidence to show that MHC matters in dogs. There's not really much evidence at all. I would be concentrating on keeping the inbreeding low generally. And as I was saying, the Embark test, it's less important how inbred your dog is than how related it is to the dog you're going to breed it with. And everything you need to know, you can see on a regular pedigree. So as long as you stick to that rule of trying to keep the pedigree, have all different dogs on it, then you won't be in too much trouble, I would say. Now, of course, there are things that pop up from time to time, and that Schnauzer one was one of those cases where actually it's a geneticist nightmare because the litter was so outbred, it was just ridiculous. You know, you have one parent from Argentina and another parent from USA or somewhere, and suddenly you've got these puppies that have got this disorder. You know, it's really unfortunate that you can't control everything. What is the significance of DLA haplotypes in purebred dog breeding? That's the same one. MHC, DLA, those are the same thing. So I just answered that one. When researching dogs, 3 generations where I had half sibling with epilepsy. Should we worry about it being hereditary in that line? Depends if it was the same sire, I guess. Not really certain from the question. I'm sorry. That goes back to the beginning where does it manifest the same way? Is it the same breed? Is it the same family? Are you seeing it multiple times from the same kinds of breeding? Epilepsy has been like something that geneticists have been working with since I started doing dog genetics back in the early 2000s. And really there's only a couple of breeds where they've had any luck with it, because there's so many things that cause seizures in dogs, just a ridiculous number of things, and they're not all genetic. All right, is there a good site to go to learn about genes and coat colors? Yes, there are some good sites. Sheila Schmutz has a good site. She's in Canada, SCHMUTZ. She's the coat color queen in dogs. That's a place to go. The Little Book's a good one. There's no electronic version of that though, I don't think. I had to actually go to the library and scan it. So
thanks, Karen, who's shared in the chat the link that we can put up with respect to Schmutz coat color.
Oh, thank you for that. Yes, yeah, like Saskatoon in Canada or somewhere.
Yep.
She's a geneticist. She breeds large Munsterlanders. Under what circumstances would a dog's DNA test fail to achieve a result? That's the poor DNA quality issue. For long stock coat GSD, does the long stock coat gene carry more strongly in the male? The answer to that is no. If the long coat is carried, I'm trying to remember the chromosome now. I think it's chromosome 5, I don't want to say. And it's a recessive gene that's caused from both sides of the family. Is hemangiosarcoma an inherited disorder? Good question. I think it is partly. I had a dog, the boy dog that I showed in my slides, he died of lung cancer, but it was probably some kind of hemangiosarcoma thing. And there is a bit of it in his family. In the US, the latest research suggests that it's affected by Borrelia, which is exposure to Lyme disease. So we do have Borrelia in Australia as well. So it can be, you just don't know what role viruses and other things that screw with your DNA, what effect they have in terms of cancer. So the answer is probably, but there's not ever going to be one gene for it, I don't think, at this point. How do we receive genetic testing results and how are they interpreted for us? That's normally the laboratory usually gives you some kind of counselling along with the result. Inbreeding coefficients again, sorry, genetic test licensing again. One lab better than another again. Okay, for a complex genetic disorder, a test is only available for one gene location. How would you treat them in a breeding program? For example, Border Collie goniodysogenesis, which I believe is an eye disorder, is that right? So it depends how big of a difference that gene makes, is the answer. And it all comes back to your perception of risk and your breeding objective, your vision of a perfect breeding program. I would suggest you're better to spend your selection pressure on things that you know you can make a difference with. And then you could just give puppy owners a warning that this might happen and that there's no real way to control it. If it's complex, you can't control it with a single test. What's the difference between having a test done as a part of panel versus single gene test? It all comes down to how far you have to spread that DNA. And that's what I was talking about earlier, where you might get your signal diminished to a point where it's not very useful and you get more inaccuracies in panel tests. I have a result showing effective for ichthyosis, but no signs of the problem. How can this be classified as affected? An animal can be genetically affected but not physically affected. That's called incomplete penetrance. The first thing I would do is have that animal retested with a single gene test, unless that was a single gene test. I would then also want to understand if it has been seen in that breed before, to know if that is a test that you should be worried about in that breed. Because what can happen is that when you make a genetic test in a particular breed, it can work quite well, even if you don't have the exact correct marker, because there's a whole section of DNA that travels together. And that marker is like a barcode that captures that segment, and the mutation can be just anywhere in there. And that marker will capture it in the first breed. When you take it to another breed, that segment is all shuffled and chopped up. And so the marker may not, unless they've got the mutation that actually causes the disease, that marker might not work at all in another breed. So you need to be careful that that condition has been observed in your breed and that that marker has been associated in your breed. And that's a job for Dog WellNet. So that's where you go to find that out. If diseases such as PRCD come back as affected, what are the prospects of the dogs not actually suffering from this problem? In the case of PRCD, it's fairly slight. I think they usually do end up getting it eventually, but I've heard of one dog that didn't get it till it was like 11 or something. What would be required to research and find marker for sebaceous adenitis in Samoyeds? I had to look up what this disorder was because I didn't know it, sorry. It really comes down to how it's inherited. And if it seems to be familial, if it seems to be recessive, if it seems to be completely penetrant, then it's just a question of getting case and control samples and doing it and figuring it out. It doesn't take too long. But you need to have the will, as I was saying earlier. You need people who are willing to make it happen. Transferring tests across breeds, they're all dogs, so how can we not be able to test transfer tests across breeds?
You just answered that. That was one of my questions, so I'll interrupt. Yep. And you've just explained that with your barcode explanation.
Oh, okay. Yeah. So as you go, as you go across breeds,
you've got the exact mutation, you can't actually transfer it across breeds without getting that mix up.
Yeah, exactly. And as a researcher, it's really, really, really difficult to prove. There's probably another 10 or 20 years work in proving that your mutation does what you say it does. So in the end, what we do is we put it out there, and then people start using it, and it either works or it doesn't, and then it gets accredited in that breed or it doesn't.
So let me interrupt you there for 5 seconds, Claire. So would you then throw the baby out with the bathwater, so to speak, and not use dogs that were supposedly affected without having a complete test done? Or would you continue through and just watch what was happening?
I would tend to err on the side of caution. So my philosophy is to treat every dog that doesn't have a test as potential carrier. And if you have a dog that shows it as being affected, but doesn't show the disorder, if it's a recessive disorder, my rule of thumb is a clear dog for every test in every breeding every time.
No, but I'm not talking about dogs that have had tests really here when I ask that question. So we're talking about the dogs that are not tested for whatever syndrome they've been given a positive test or a carrying test for, that I'm asking.
I always regard a dog that's not tested as a carrier. Even though it may or may not be a carrier, if it hasn't a test, treat it as a carrier.
But what if your breed doesn't have that disease?
Oh, okay. So there's been one that's so go to Dog Wellnet and see what other people's experience has been. I had a case recently that was told to me of von Willebrand's type 1, I want to say, was turning up as a common affected condition in a particular breed. And I actually went back to the patent for that test, and when I read the patent, I was a bit horrified, because the patent shows that actually it's not even a coding mutation, it's just a silent mutation in the gene. So I just don't, in the original patent, they used Dobermans for this particular disorder, and it seemed to have some impact in them. There was some research work that came out of the UK, and they came up with a genetic severity index. And so, but it's a good sort of rule of, it's a good sort of thing to think about is, as a breeder, or as a breed club or whatever, have you seen it in the breed? Is there a clinical phenotype? It's like the CEA test in toddlers. People have been doing it for years. It costs us hundreds of dollars. It used to cost $400 to do the test. And the recent research shows that it doesn't make them go blind anyway. So why would you bother? I mean, really, if it has no clinical outcome, it's not worth bothering with. You really need to be careful about spending your selection pressure wisely. And as I was just saying, not every test works in every breed. DM doesn't work in every breed. Other dogs have some compensating mutations somewhere that stop it from doing anything. So, you don't know how a gene test is going to behave in a new breed. So we've recently, we've been doing some research on a cerebellar abiotrophy in Kelpies. And we didn't release it, but the research that we did was released as a test to the Kelpie people. And we only had Kelpies in our research. but someone messaged me and said, oh, I've got this border collie and it's got all these signs, and can you make us a test? And I said, well, why don't we try the Kelpie tests first and see if they work? And if they don't work, then we can have a look at making you a border collie test. Well, this person ran the Kelpie test, and sure enough, one of our Kelpie tests perfectly described what was going on in their working border collies. So that saved us a whole lot of research, and they had something ready to go. So sometimes it will work in another breed, but, you know, I was talking to another breed club recently, and they had a juvenile onset Addison's disease. And I said, look, why don't you just try the Toller marker and see if it works? And in that case, it didn't work, but it's worth a look, because then if it does work, you've got something ready to go and you can use it straight away. Whereas if it doesn't work, then you've got to do the research to find it. So it's worth having a look, but if you're getting things that are turning up positive and you didn't ask for them and you've never seen the disorder in the breed, I'd just say, don't worry about it. Just don't worry about it. It's not worth it. Not worth it to waste your anxiety on it, I would say.
Now, 9:10, Claire. How are we going on those Slido questions?
I've got a few more yet. Well, I'm happy to stick around for a little bit longer to go through them, but people can go, of course, if they want to. What, the Zoom doesn't usually cut off, I don't think, or does it?
Well, I booked us extra time anyhow, and it all comes at the same price.
Yeah, so if people want, I'm happy to keep going.
Thank you.
Yeah, I mean, everyone, if you want to leave, feel free to leave. But if you want to stay, you're welcome to stay too. All right, so, oh, now I'm lost. Does a litter have the same DNA profile? No, they don't. All members of a puppy of a litter will be different because they're not identical twins. Just like you're different than your brothers and sisters. What were my thoughts on genes for fear? We do a lot of research on anxiety, and anxiety is a complex disorder. No, we shouldn't say disorder, a complex trait, because we need to have some anxiety or we run in front of cars and things. So anxiety is useful for survival, but you don't want too much of it, I guess. Why do some companies offer tests for diseases in my breed? Now, the reason panel tests offer diseases that are not in your breed is because they use the same panel for every dog breed. And what some of them seem to be doing is they've got a test for like a husky, and that was put on there for huskies, and you get a corgi that shows up as being positive. And so now they offer it in corgis. That's why they do it. And I would say ignore them. And, you know, from an ANKC perspective, I'd be pushing for a filtered test report that can be given to puppy buyers that only lists tests that are relevant to that breed, because even if you want the rest for a heads up to see how it goes, I really think we need to be careful about how we report our testing results going forward, because a lot of these things are not 100% accurate, and we could be making things much harder for ourselves than they need to be.
The reality is that with your husky corgi example, it really hasn't been validated for a corgi.
No, exactly.
So it's sort of naughty to be reporting it, really.
Well, but most things are not validated for most things, reality, you know.
So, Margaret, would you like fries with that burger or not?
It's just that, you know, I'm used to working with chemicals that are nice and black and white, so you can genuinely go for things and, you know, 98% confidence limits. Right. And how's the list going, Claire?
Is it possible to make a reliable DNA test when the test was developed on one sick dog? Sometimes, yes, but it's harder. I was recently asked to review a paper where they had, you know, it was a panda, and it had cataracts, and they'd done some candidate genes, and they'd found a mutation in one of their candidate genes, so they had determined that this panda, that this gene caused this cataracts in this panda, and it's just like, no, probably not. So I would say treat with extreme caution. X-linked autosomal, well, if disorders are X-linked, then they're not autosomal, because autosomes mean not X-linked. If a bitch is a carrier of an X-linked condition, then it doesn't matter what the dog has, because it affects the boys depending on what the bitch has. It's highlighted bitch for me because it thinks it's a swear word. Slido asked me to moderate those. If a bitch has been mated by 2 dogs and pups need to be tested for parentage, what age can the pup? I think we covered that one. Right, just general things. Oh, so a dog has tested positive to black hair follicular dysplasia. The DNA report states positive. May express phenotype. We should not use to breed with. Now, this is the one I actually said on that made me stop on my own breed one, because this is unpublished work. There is nothing reported on it so far as I can tell in the research or public domain. So I don't know where this test has come from. I can only presume it's an in-house secret test that they're not sharing with anybody. And I would say if your dog looks fine, black hair follicular dysplasia, what it means is the black hair all falls out. If your dog has black hair and it's not all falling out, I would forget about it. Forget about it.
Just a question for you, and it's me interrupting again. Often my puppies are born with black hair, and that all falls out when they get to a certain age. So is that related to what you're talking about? They don't go bald, they just lose their black hairs.
What colour do they end up?
Somewhere between, let's say, brown and a reddy fawn colour, or a light fawn colour, maybe like your breed of dog. Yeah.
Okay, so the A locus, no, that's a different thing. So typically on the A locus, which codes for shades from like the wolf colour, it's called the agouti locus, from the wolf colour through to fawn or yellow, what's called a yellow sable, like a dingo is a yellow sable, through to black and tan, through to solid black. In the dogs that have the yellow sable, they're often much darker when they're young than when they're older. So if you look at baby beagles, for example, baby beagles are often quite dark, whereas as they age, they get lighter and lighter and lighter. And it's the same in our breed. In our breed, we have some dogs that are sabled rather than red because of the MC1RE locus. And in those dogs, you can see it in them when they're puppies, but pretty much by the time they go to their new home, they just look red. And so that's not, the hair's not falling out so much as fades out. It's a different thing. It's not the same thing.
Oh God, mine fall out. I see it in the whelping box. It completely falls out. By the time their eyes are open, you see the black hairs falling out in the whelping box. It's hilarious. Like, oh, there you go. There's all your black hairs gone. We don't need black hairs in our breed because it's, you know, a sight hound, so they can be any colour. But it's very interesting. It always fascinated me that all the black hairs would fall out. Doesn't really matter. But yeah, I was wondering whether that was what you were talking about or not.
Yeah, no, that one's a different. No, it's more like the dogs actually lose all the hair on their ears. They look like they've got mange, pretty much. So if you had a dog that had that, you'd know it, I think. So, okay, so Margaret, what do you want to do? We've lost her.
Yeah, sorry, my bitch decided she wanted. I'm in that puppy room, as you might hear, and my girl wanted out. Do you want to go across? There are some questions in the chat now.
Yep, I'll have a look at some questions in the chat, see which ones we didn't cover.
Right. So let's start with a more general breeding question. One here says, if you have a line-bred dog, can you breed away from it using outcrosses?
Yeah. Doesn't matter how. If you had a dog that was 100% inbred, if you bred it to a dog that wasn't related to it, all of that inbreeding is gone in the next generation. Disappears entirely.
Yep.
That's why I'm saying that the embark thing's kind of irrelevant, because how inbred the dog is it's not relevant to anything. If the dog's healthy, it doesn't matter. What does matter is how related it is to the next dog that you breed it with.
Yes.
So there's a massive misnomer in dog breeding about the fact you can put a heavily line-bred dog over an outbred dog and your traits will carry through. This is the whole history of purebred dog breeding in a lot of ways. Is that a complete misnomer, Claire, or is that something that does actually ring true?
If you have dogs, it's like that picture I showed with the German Shepherd and the Basset Hound, right? The puppies from that litter, the two little brown dogs, look kind of even. So with line breeding, you do tend to get litters that are more even because there's less genetic variation there. But if you actually want to go somewhere in your breeding program, what you do is you breed yourself into a corner where there's nowhere to go. And I guess that's if you want someone to know that your dogs come from your kennel, but you know, if you're branding, I guess that's what people have done in the past is they've done branding. It's okay, but then if you have a disease that you have to work with, or if you have suddenly got to put a nose on your pug or something, you can't do that because there's no genetic variation left. So you can do it that way, and yes, there's a bit less genetic variation, but you can get more even litters by breeding into families that have those same characteristics too, right? So yeah, I think there's more than one way to do it, and generally the healthier way is to not completely outbreed, in that you don't have to crossbreed, but use less related individuals from those that are available.
We haven't had any questions about crossbreeding, and crossbreeding because breeds are actually about to die out or have so many diseases. This country, we can't do what they call the Outcross project. What's your view on some of the European countries that are allowing the outcross projects and then breeding back into their lines of dogs to get some genetic health back again?
I hate to say this, Marcus, we've got a few questions still outstanding in our chat list before we head down that.
I'll shut up.
Okay, so I'll come back to that later.
Yeah.
All right, so
I've lost my place, but I did have some good chat questions. Now, I've lost my place. So we're all being encouraged to go down we've got the line-bred European dog, the outcross breeding, we understand. Here's one. A vet thinks that one of my older girls has the start of cataracts at 9, yet her DNA test for HC is clear. So are both parents and grandparents. Would I assume the vet is wrong?
I think there's more than one kind of cataract. And I think that cataracts in old dogs are not uncommon. I think it's the ones in the young dogs you have to watch out for.
Yes. juvenile issues.
Yeah.
People are asking about best testing laboratories in Australia. Sorry about my puppies. But
pay attention to me.
their mother's waiting for her supper and won't hop in with them. The question about labs in Australia, we just send our genetic material overseas, yes?
There are, I'm trying to think, there's at least 2 in Australia, I think. There's a little one in Sydney that's just started up, but we don't have a lot of option here. But
Yeah.
it's not hard to send things overseas either, so you just do what's best for you.
Yep, and COVID isn't causing an issue for that in any way? There's no problems currently?
I think it was in the US for a while, it was slowing things down, but I think they're ramping back up again. But who knows what COVID's going to do to anything.
Yep. Okay. Now, so, and in fact, we will need to go overseas for some of those single gene tests. Yes?
Possibly, yeah.
Yep. So where can we put our single gene tests in for our dogs, right? So
the Dog WellNet has a list for all the test providers for each test. So if you put in your breed, it will tell you which tests you should have, and it will also tell you which labs offer those tests.
Right. Okay. Now, what else? Oh, your view on breeding a dog and a bitch not related. Well, you've covered that.
It's hard to do. I mean, it's not always possible depending on the size of your breed. Like in our breed, it's quite hard because it's only a small breed.
Well, and you can get a good COI in, it's all right, my husband is not listening to my direction. She wants a supper, please. You can have a 0 COI at 5 generations, but 10 generations quite significantly different, but not ridiculously. Yeah.
I don't worry too much about the long ago inbreeding.
Yep. Now I'm again back to so breeding them not related, good information. We will endeavour to put up the links for the little publication, and also the woman whose name I can't remember that started with an S.
Sheila Schmutz.
Yep, that's it. Thanks, Karen. I think that we might be linkage versus direct testing. I've got no idea what's meant.
So when you have a test, you should know whether it's a linked marker or whether it's the mutation that causes the disease. But that's the point I was saying about it's really, really, really difficult to be 100% certain that you've nailed it in terms of having the exact right marker that causes the disease. So if it's a marker that's nearby, that's linkage, and that means they can get disconnected through different breeds, or just in the same breed over time, they can get disconnected depending how close they are. So it's good to know if it's the mutation that causes the disease or if it's just a marker that's near it.
And the paper will tell us that, the original publication?
Even when we publish, we still say the putative mutation. We don't say the mutation. We're not allowed to. We've lost Margaret. She's gone to feed a dog.
Yes, you've lost me.
Are leg breaks in Italian Greyhounds genetic? I don't know. Maybe. I don't know, because we're working on one in Bull Terriers that they have broken legs that are genetic, I believe.
Right. And sorry, just back to that, because I had to get up and pick up the squeaky puppy. Back to the linkage versus direct testing. In terms of validating that test over time, for some of those, will it just be a case of collecting the data?
Most of the tests think that they're mutation tests these days. There aren't many linked marker tests. People think that it's the mutation, but they may not know it's the mutation. And so time will tell. All these things are revealed eventually. That's a very cute puppy.
Yeah, it's a very noisy puppy. Right. How deleterious are genetic bottlenecks for the overall genetic health of a breed? This is in relation to Dobermans.
Or any breed, really. They're not ideal. But, you know, I'm not very popular always in amongst the geneticists because I tend to be quite pragmatic about these things. And my view is that in evolution, animals didn't arrive on Earth with an NE of 50, an effective population size of 50. That's not the way it happened. Animals evolved with a few animals being separated off and becoming a new breed or species or whatever. So I think that they're relatively resilient, more resilient than people give them credit for getting through a bottleneck and coming out the other side okay. But, you know, it depends how many bad genes are in there. And if you can always get a bit out of that bottleneck, then that's always a good thing to do. But in terms of the way things are going globally, and there was a question, I think Marcus asked this question, or someone asked the question about the European people wanting to index dogs in because of the Dutch Kennel Club wants to index dogs in. And I do think that is going to happen no matter what we think about it. And then we have to decide what we as a community want to do with that. And personally, by the time it's third cross back in, you wouldn't know it's not there anyway. I think if someone's dedicated enough to index something in, then good on them. But, you know, it's up to you whether you want to use that dog in your breeding program or not. I think as long as they're clearly identified, I don't have a problem with it, personally.
Well, we can't at the moment clearly do any of that. This is not a question for my breed of dog. This is just a general question for the breeds of dogs that are looking not fantastic at this point in time as far as our ANKC goes here in this country. So it's a very broad question that I'm putting out there for some of the breeds of dogs that are, let's say, looking like they're ailing as far as this goes.
Yeah.
So, you know, we all love whatever breed of dog that we breed. So my real question is, what will it take for those people that breed these breeds of dogs in this country, maybe at least, to be able to access that kind of thing? How bad is it going to get before they can at least, you know, cling to the shirt tails of the people that are overseas? And probably this is like a rhetoric question, I suppose, before we can start to benefit is probably not the right word, I don't know, from what's happening for some of those countries that are being a bit more progressive from what we're doing with our closed stud books at this point in time, because we're sitting still with the eugenics of, you know, the 1900s here, and for some of the breeds, it's a really big worry.
Yeah.
Yeah, so probably not a question you can really answer outright because you don't know what's going to happen. But what's your opinion on that for some of those breeds?
So generally my opinion is that I personally, I think we're too precious. Like, but that's just me. Because at the end of the day, if a dog looks like it's meant to and breeds relatively true, that's the way all of our breeds started, right? I found a book in a bookstore. Where was I? Albury or somewhere, and it was on dog showing in the 1930s, and actually I had classes for crossbred dogs in the dog shows back then.
Oh, that's because it was Albury. I can say that because I used to live there.
At the latest International Dog Health Workshop, which was in London in 2018, the general consensus in the room was that that was the way the kennel clubs were going. And I think it's a positive step, but I do think that if people want to go that route, it actually takes some level of dedication to make it work. And I think that most people won't have that level of dedication, but I think it's okay to have that option there for people that do want to do that. My concern is that those dogs wouldn't have a meaningful impact on the population because people wouldn't use them. But if people do end up in a hole, like I made a comment about pug noses, but if we go the way that Europe's going and you don't have enough genetic variation in your breed to be allowed to continue breeding according to the rules that are made by lawyers, then there's going to be no other option than to do some kind of outcrossing. Otherwise, we'll be here for 30 years trying to make it happen.
But you did make a very good point when you first started to talk about the topic, insofar as you mentioned that it needed to be noted, that the dogs needed to be identified, because we had the scenario way back with the bobtailed boxers, yeah, and the
like the Recessive Dalmatians,
and it was never identified.
Oh, I see. Yeah.
And that was the issue. It was the fact that it happened without consultation with the international community, because it's all very well for the Netherlands to be doing whatever they do, but unless they identify the pedigrees, then...
I think it's most important that the animals themselves are identified. The same thing happens in horses, right? With quarter horses and so forth, they index them back in, and warm bloods and things like that. I think it's very common in other species to index animals in, but the animals are always clearly identified as where they are in the breeding process. So whether they're a first cross, second cross, third cross. Honestly, by the time they're a third cross, you wouldn't know they're any different than the regular one. And I've done research with the Recessive Dalmatians, and they're genetically, they're no different than the regular ones. So
genetically,
yeah, from a DNA perspective, they look precisely the same. So it's not. I think people worry too much. Generally, people worry too much. So don't worry so much.
Oh well, look,
we have to worry because we're bound by certain things here.
Yeah.
The AAC is the bounding thing that we have, which is fine. But you know, one of the amazing things about the canine genome is the diversity that it has. Is there any other species that has this diversity, in your opinion?
So what you're talking about is not that the genome is diverse, but that the dogs look very different from one another. Is that what you mean?
Yeah, that's actually what I mean. Does that mean the genome is diverse, or what does that actually mean? Why do we have...
Well, actually, it just means that we have... selection is very powerful. And when we select for something and we get an effect, we keep selecting for it. And the result of that is our dog breeds. And actually, the number of genes involved in making the way our dogs look isn't that many, to be honest.
So what about livestock? What about cats? What about all of those things? Do they have that genetic diversity? Pigs, whatever.
Yeah,
They don't, do they?
Well, they do. They're similar. They're not dissimilar. It's just a question of how much effort has gone into the way they look, as opposed to whether they have juicy chops or something, right?
The variables are different.
Yeah, the breeding objectives are different.
Yep.
So, you know, with dogs, I think there was a lot of incentive to make them look different than their wild cousins. So that promoted all the different colors and shapes and forms. And also people just love things that look a bit different, right? So, you know, the puppy in the litter that's odd, that's always the first one people pick, right? So they want something that's a little bit different.
Yep.
And I think that all of our breeds have come from people wanting to promote something that looked a little bit different and fostering that.
So you're saying this is a result of the human genome rather than the dog genome, maybe?
Yeah, no, well, yeah. It's our desire to, you know, manipulate and control things for our pleasure.
And of course, the world has changed. So functionality was often fairly important in a lot of these dog breeds historically that we don't have. I mean, you know, you think of all the ratting terriers and those sort of roles, genuine functional roles that the dogs played in our world.
Yeah.
Well, I think that we've probably come to the end of the questions, and it's perhaps time to remove some anxiety with a glass of wine. Thank you so much, Claire, for your contribution.
Thank you for those of you that asked questions. It was they were interesting. I really enjoyed them. Thank you.
And yes, it's been great that you were able to, and we do wish that Trouble continues to make some level of improvement for you and that science can help out.
Yeah.
Okay.
Thanks, Margaret.
Thank you so much,
Thank you,
Claire.
everybody. See ya.
Bye-bye.
Bye.
Thanks, everybody, for attending.
Bye-bye.
Bye.